for Sequencing and Introduction to HALE. This video, for video number 4, we'll be focusing on next-generation sequencing technology, specifically in the informatics, and this would be in line with the 2021 virtual workshop on statistical genetics methods for human complex traits, hosted by the Institute for Behavioral Genetics, University of Colorado Boulder. My name is Kumar Virabin, and welcome to this video. Objectives for this video would be to understand the overview of next-generation DNA sequencing methods, specifically in the bioinformatics, where to obtain an overview of the bioinformatics involved after obtaining the sequencing reads. So I'd like to preface this by just mentioning that it's been a while since I had to code a lot of this pipeline. It's been probably almost half a decade right now since my PhD, and so there are a few nuances or technology advances that I try to update myself with Laura Gautier from the GTK team, which is why I thought this is like a great segue from our previous knowledge of how sequencing works, where from the tube you would put it into the sequencer, and from the sequencer, after all that picture taking that we were going through in our last video, you would have the reads being spit out by your sequencer in FASTQ files. These are .fastq files, as the note written down there. GTK is a tool that you can use that I'll be introducing to you, that you would then process your FASTQ files into BAM, SAM, or CRAM files. SAM would be the non-binary version of the BAM files, something alignment map files, and CRAM would be the smaller version that is commonly used right now, especially at the Broad. And then, so GTK would process FASTQ. The intermediary files would be the BAM, SAM, or CRAM files that you can save that has a smaller size and easier portability. And finally, with using GTK, you would produce variant-ready VCF files. So when it comes to next generation alignment and assembly, if we think of traditional methods of just doing alignments, just like using NCBI tool like BLAST or BLAT, it can be too expensive for a huge number of short reads. Remember, these reads are about 99 nucleotide longs. That's it. New tools apply established alignment algorithms and new algorithms like the BWA, which works wonderfully well, the Burroughs-Wheeler alignment. And some older methods of sequencing uses just equality values for alignment, such as Solexa, which is what Illumina is right now, shrimp on solid. So the MAC, the mapping quality, would be one of the main things that we use for alignment. De novo assembly can be challenging, but it works really well with Illumina reads, which is why a lot of the other NGS methods that I alluded to in video number 2 really just has been phased out entirely, just because the Illumina method is so much easier because it's paired-ended reads. It's kind of like, if I see this here, I will see this the other side, and if I don't, I would have lower confidence that that's really what I'm calling. So the GTK pipeline can be delineated into these 3 different phases. This, I'm glad that I recently found out, was actually created, I want to say the early 2000s when GTK started, because when GTK first started, it was created by a bunch of software engineers, and they essentially just said, I have all of these tools, and all of these tools can be used whenever, but as geneticists, we like having recipe books. And so a good friend of mine who's one of the she's the lead for the engagement and support for GTK, and now on Terra, worked on this, just how do I use just the things that I need to use and just phase it out into however many phases that I need, this is something that she's very proud of, and I'm very proud that she did it, because I would not have understood how to use GTK very well and probably still be in my PhD program. So when it comes to the analysis, GTK is one of the major tools that we use, where GTK is a broad unified genotyping collar. It can be used for multiple samples, and it's technology aware of whatever SNPs that you use, and also it's an indel collar as well. Which is later we'll go through what kind of collar you can use. The unified GTK collar, as I mentioned, can be used for multiple samples, and even HALE can be used within, actually, HALE can be used probably in phase 2 when you do joint calling. But I'll go through what these means in a few moments. So let's start with phase 1. So in phase one, you would have raw reads, the FASTQ files that enter into the GTK pipeline in phase one. You use non-GTK tools like collect ATEST metrics in order to generate your quality metrics, and Picard tools in order to mark whichever pieces which are duplicate. Honestly, I also use Picard tools to also obtain some quality metrics, like what your reads are, how your reads look like in your FASTQ, just so that you could do some pre-QC here. And honestly, the data processing takes a while if you don't have a pipeline ready for maybe whole genomes and whole exomes. If you want to see recommendations that we've used for the COVID-19 Host Genetics Initiative, I wrote up something really cool for whole exome and whole genome sequencing with Laura Gauthier, the person I was mentioning from GTK, who works on the bioinformatics, that wrote up for me a lot of recommendations on what tool to use, what flags to use, and what do you have to be on the lookout for things which are good or bad in this document, where the link would be available for you in the PowerPoint that I'll be sharing later. So anyway, back to GTK and getting the metrics and just seeing what works and what doesn't. You would then do your alignment and local realignment using the Burroughs-Wheeler aligner tool, BWA-MEM, and this is the version that's the most current version as of the writing for this Google Doc here. You would then do base quality recalibration, which is essentially just taking the base quality that you have per base and recalibrating it to whatever you have in the entirety of your data set, or what you have as the multiple reads that you then align on top of to each other. You would then call on to, like, this would be this sequence, because remember, each of these polonies could be duplicates of each other. And so when you have duplicates, multiple duplicates of that, when it comes to transcriptomes, you can infer many things. But when it comes to exomes and genomes, they would just increase your confidence that that is truly that nucleotide, which is what the base recalibrator does, specifically base recalibrator and apply BQSR. This would then reduce the number of reads because you're throwing out reads which just, look a little suspicious, some duplicates that you don't really need, such as in duplicates, you would look at orphan alignments, things which don't align with any other pairs, because remember, in sequencing by synthesis, you had pair under reads. When one thing occurs, the other thing should have an overlap. If you don't see it, you probably have to throw it away. Secondly, if you have unmapped mates of duplicate orphan reads. So you would have reads that are duplicates but don't have the duplicates of those duplicates. I know, like duplicate, duplicate, duplicate. But if you think about it, if you have 7 of sequence A1, you should have 7 of sequence A2, right? A1 being the, one pair and A2 being the second pair. But just say if you have 7 of A1 and only 5 of A2, you can just throw out the 2 from A1, which is kind of like the leftover. But there's a few quality metrics that you probably have to talk to a bioinformatician before you decide. And really, QC is really important, even at this upstream method here. I don't talk about a lot of the bench QC because you don't really do this. But we don't really do this, but other people do. But for this course, not truly important. This one I'm going on and on, just like letting you know and reiterating QC, extremely important for every application, practically in your life. And so moving on to phase 2. So in phase 2 is the variant discovery and genotyping. So you would have each sample from the FASTQ forward and reverse, or 1 and 2, that you would now have one BAM file. And then sample 2, you'll have another BAM file. Sample 3, you'll have another BAM file, or SAM or CRAM, whatever you prefer, right? So upon getting this, you would use the haplotype color in order to find out what your SNPs, indels, and structural variations are. And the resulting GVCF can be used for your joint discovery workflow that would be in phase 3. So I'd like to also mention indels are like, maybe up to 10 nucleotides long at most, I think. I don't really work with indels as much. I just know like indels are not called very well using haplotype color. If you'd like larger structural variations, happy to point you to good references, but many things coming out from Michael Tarkowski's group at MGH can point you to a lot of really good resources and tools that they've used in order to call out structural variations in this pipeline, and on top of that for the analysis for structural variants. And so this brings me to the final phase for GTK, which is the integrative analysis, where, if you recall, each of these samples are per sample BAMs, and right now, per sample GVCFs. So we would want to join them. So the GTK tool that you would use would be reblock GVCFs, which you'd have to take individual VCFs and join them together. So just say you've already joined them and you have like maybe 100 whole genomes in one VCF file. And it's really important to do this because it's really easy to run your analysis if you have one VCF file, as opposed to having 100 multiple GVCF files which you'll run. So back to the thought of like, if you've already done 100, right? And that's your data freeze. So your second data freeze comes in. And when your second data freeze comes in, you have another 100. Following GTK, you would have to go back to the individual larger size format of BAM or VCF files, run through until you reach reblock GVCF, and then you'd have to reblock them again. So the 100 that you did before, you'd have to do 1, 2, 3, 4 until 100 plus the 100 that you're getting right now. So it's kind of like a little bit of it's a little bit annoying I wish I could speak in Cantonese right now because there's a word that you would use for this. But anyways, it's just like a little troublesome. So using the Hail VCF combiner, if you already have the one file for the 100 VCFs, you can just use the VCF combiner to add the other 100 directly into it post phase 2. So you don't have to do one by one all over again. Anyway, that's a quick plug as well onto the VCF combiner, which is quite honestly one of our most popular tools and most well-requested tool at ASHG in 2018. 2018 and 2019, two of the last ASHGs that I got to go to representing Hail. So then you would have to do the variant call filtering, which would be the last 3 oranges over here. After you have your raw variants, you would join them. You would then have to see how good they are after joining by using genomics DB import, variant recalibrator, and finally apply VQSR. VQSR is a metric that you can use in order to identify which are good reads or good variants that were called or not. And finally, going back and forth between each of these QC steps, you would have analysis variance, which is the end of this video. And the learning outcomes for this would be you understand the overview of next generation DNA sequencing, where you have an overview of the bioinformatics involved after obtaining the FASTQ files, the sequencing reads. So video for next generation sequencing, informatics. My name is Kumar Veerapan. If you have any questions, feel free to send me a Slack message or send me an email at veerapan@broadinstitute.org. Thank you.